Hello everyone, and welcome to a new and somewhat different Atomic Ecology Meets. Last week I had the wonderful invitation from Simon Johnston to head over to Edinburgh for the annual scientific meeting of the Freshwater Biological Association and talk to them a bit about stable isotopes, what I've been doing with them, what you can do with them, what they should be doing with them, and what atomic ecology is doing to connect all of these pieces together. And I gave a 30-minute keynote address to their conference, and I decided it might be fun to record it now that I'm back home and share it with everybody else. So if you've got 20-something minutes and you're interested, stick around. We're going to cover this in 3 parts. We're going to start with some very light introductory theory and thoughts. Then I'm going to talk about a couple of nice real-world applications that I've been involved in. And then, well, because I have to pay for my own supper around here, a short little sales pitch really around my role with atomic ecology and how I feel it can benefit your own work. So we're going to start this story, like many, many good stories, on a sunny day long, long ago. And while this story starts long, long ago, this sunny day was actually last Wednesday in early June when I was walking through Glasgow, the leafy suburbs of Glasgow, looking for a very important plaque. And I found it just around the corner from somewhere called Isotope Lane. And the plaque refers to an important dinner party that happened over 100 years ago, where Frederick Soddy and a dear friend of his, or so I'm told, Margaret Todd and others were having dinner and discussing this chemical, physical properties of some of Soddy's work. And either Soddy or Margaret Todd came up with the term isotopes as a good name to reference what we now know of as an isotope. But before that, we didn't have the word. So Soddy got a Nobel Prize for his work, and this plaque commemorates all of that. And I was very happy to find it, take a picture with it, and continue on my merry way. So what are these isotopes that Frederick Soddy and his friends were interested in? Well, all of us, if you're listening to this, I'm sure you're aware of this concept of a periodic table of elements, where we can get all these elements and divide them up or catalog them based on their physical and their chemical properties in terms of their atomic mass. But isotopes are nature's way of kicking back against the machine. Because while we like to put things in these neat boxes, nature doesn't work that way. And what we see here in front of you are 2 separate isotopes of carbon. Carbon, we know, is the basis for all living matter on life, on Earth at least. And isotopes record a slight variation within carbon. And essentially, we're looking at 2 different atoms here. Up above, carbon-12, 6 protons, 6 neutrons. Down below, carbon-13, 1 extra neutron. Makes the same bonds, does the same things. It's still carbon, but it's got a slightly higher atomic mass. And this slight difference of atomic mass is really important for 2 reasons. First of all, we can separate out these isotopes in isotope ratio mass spectrometers. So we can ascertain what the relative abundance of each of these in any piece of material we put into our mass spec. Secondly, they behave, I told you they behave the same way. They do almost behave the same way. Heavier isotopes make slightly stronger bonds, which means that lighter isotopes are a little bit more active and reactive. And that means as we go through any biological, chemical, or physical process, the lighter isotopes will be not preferred, but they will be a bit more active. And here we can see a consequence of that. And this is a estimation of the relative abundance of light -30s and heavy -10s isotopes or isotope ratios in plants. And what we see is this bimodal distribution, whereby C3 plants have one range of values and C4 plants have another range of values. And if I had atmospheric CO2, it would sit around about there, around about -8. And essentially what we're recording here is that the C4 plants are more similar to the atmospheric CO2. They're more efficiently using CO2. The key part of this, or the key take home from this, is not that C3 and C4 plants are doing slightly different things. Rather, it's that isotopes are a really good way of quantifying and measuring these different processes that we see, that we're interested in as ecologists. Remembering, though, that we are talking to, or at least we were last week, the membership of the Freshwater Biological Association. So how does this relate to freshwaters? Well, if we start combining a couple of isotopes together, and here we're looking at a biplot where carbon on the x-axis and nitrogen on the y-axis, we can tell an awful lot about how a system is functioning. And this figure here by Jones and Gray several years ago is actually, anybody who's taken my courses is well aware of this, but is one of my favorite figures of all time because it is so informative. And what this figure is an isotopic representation of this. This is beautiful Loch Lomond in the centre of Scotland, a little about an hour's drive north of Glasgow, where I was very, very fortunate to be able to visit my good friend Chris Harrod, who works around about there somewhere in the Scene laboratories as part of their field station for the University of Glasgow. And many hill walkers and hikers view Loch Lomond like this. Lots of us who are interested in freshwater biology are well used to looking at lakes and rivers and trying to figure out what on earth is happening under the water. And for my money at least, or my opinion at least, stable isotopes are one of the best ways of figuring that out. And now we're going to to do this, we're going to look at that figure in a bit more detail. So, orienting ourselves, we've got carbon on the x-axis, nitrogen on the y-axis. We can look at primary producers, the base of the whole food chain. In fact, how did this data come about? This data came about by researchers going out, sampling bits of everything that they could find in the lake, all the different species of fish, different invertebrate, zooplankton, different things that could be fueling the food web, and just analysing them all, running through a mass spectrometer to get a picture of what the food web in this lake looks like. And we can see here, I'm going to change to my laser pointer here. There we go. Right, we've got phytoplankton. We move up a trophic level, zooplankton. So we see here, similar to the C3 and C4 plants, phytoplankton and terrestrial leaf litter, terrestrial plant material, have two separate carbon isotope values, tracing distinct sources of primary production. These carbon values, though, are conserved between predator and prey. So if we look at zooplankton, they've got similar carbon values to phytoplankton. We look at eels or Arctic char. They're feeding pelagically. The Arctic char here are feeding on zooplankton. We've got this range of a pelagic food chain within our lake. We also have a benthic food chain within our lake, starting here with terrestrial plant material, macroinvertebrates, some small littoral fishes, trout parr, sticklebacks, pike, which are a predatory littoral piscivore, feeding on the shallow water species. We've got some bigger trout here. apex predator in the system are these big ferox trout, and they're integrating pelagic and benthic resources. The other brown trout are doing something similar. If you look at the range of nitrogen values, in fact, for these other brown trout, we've got some of them up here, I think they're behaving, feeding like ferox trout, and others sitting down here feeding like sticklebacks. So we've got multiple trophic levels within our population of trout. Look at these minnows. We've got some minnows very closely associated with zooplankton. We've got other minnows much more closely associated, feeding like sticklebacks, much more closely associated with benthic macroinvertebrates. Now, we've got Atlantic salmon. These Atlantic salmon have a totally different value. These are adult Atlantic salmon that were migrating back up through the lakes, heading back to spawn. Their isotope ratios reflect marine foraging, feeding way out at sea. We've got this, again, totally different values here on the chironomids. These are deep water chironomids feeding on a microbial loop. So they've got very different source of carbon again. But just see what we've been able to do just by collecting these samples, visualizing the data, and starting to tease apart some of the processes happening within our lake. We can identify distinct production pathways, even incorporating microbial production. We can look at consumer ecology. We can look at organisms, or we can estimate diet. Are species a generalist or a specialist? What position are they at in the food chain? Are there any migrants? Are all the organisms we're sampling resident here? Or have some of them arrived or been introduced from somewhere else? I first came across this paper and this tool as a PhD student, and it lit something in me that hasn't gone out yet. And I'm still fascinated by how we can apply this tool and infer all sorts of ecological mechanisms. And this has led me, a merry dance, some might say, has led me on a very interesting career. I've got to visit all sorts of places around the world. I didn't get to visit all of these places, but I got to visit some of them. Starting out in Ireland, doing work with cyprinids, then moving to other places in Northern Europe and Finland, doing work on salmonids, and then moved onwards to Canada, where I ran a research lab there and worked on a whole variety of different types of organisms. And for all of these organisms, we're able to answer similar types of questions around trophic interactions, ecosystem function. What role are they playing in a system? And how does the environment around a system regulate the type of ecological roles that system can support? Isotopes give us a tool with which we can quantify all of this. And that's why I love them, and that's why I think they're the best technique out there. I want to run through a couple of case studies, a couple of examples from my own work to highlight some of these applications. The first of these stories starts in beautiful Finnish Lapland, way up north of the Arctic Circle. This is Kilpisjärvi. This is Kilpisjärvi Lake out here. That's a little hut. Very similar. We didn't stay in that one, but very similar to the types of accommodation we were staying in while we were working up here during the summer. I'm going to show you data from the summer, but delve into my Google Scholar and you will find other work in there from less hospitable periods of time. Yeah. What are we dealing with up here? We're dealing with some of the most beautiful lakes imaginable, or pronounceable even. This is Lake Kilpisjärvi, Järvi being the Finnish word for lake. Northern Finland, over there we're looking into Norway. Sweden is on that side of the lake. This lake, crystal clear water, Secchi depth well below 10 meters, cold water, full of beautiful salmonid fishes, be they char, trout, whitefish, and grayling are some of the examples here. I arrived here, not freshly minted PhD student, give or take, and was just simply blown away by the variety of these ecosystems, their natural beauty, and their abundance, the abundance of fish that we find in these systems. But these environments are changing. Yeah? We know that climate change is happening. We know that it's has its greatest effect in northern latitudes. And this was really what was fueled or funded, at least, the postdoc project that I worked on up there, was trying to understand how a changing climate is going to affect the lakes and the lake ecosystems. We studied this I worked very closely under Kimmo Kahalainen, who was a wonderful, or is in fact, a wonderful aquatic ecologist, limnologist, fish biologist, based up in northern Finland. And over 3 years, Kimmo and myself and a motley crew of summer students and master's students worked sampling around about 30 lakes, spanning a 500-kilometre latitudinal gradient, 500-metre altitudinal gradient, from up here in Kilpisjärvi to where the Tornion-Munionjoki drains into the Gulf of Bothnia, the top of the Baltic Sea. And that sampling encompassed around about 3-degree range of mean annual temperatures, or indeed summer temperatures. So what we're doing is using this almost as a space for time study to understand what's the future going to look like, what's the future for Kilpisjärvi going to look like, and how does the functioning within the lake change as we move along that gradient. Because when we got to the southern component of this region, this is what we were met with. We were met with very different lakes, very murky, darker water lakes, full of macrophytes, and a very different fish community. Here we've got things like ruff, roach, my personal favourite, perch, pike, present throughout the range. But we've transitioned from a pure northern fish community to what really is more typical of a Central European fish community. And we were interested in what happens to the fish community. Well, we're interested in the fish community, but here I'm more so interested in the ecosystem function, the ecological function within lakes as we move from this beautiful, pristine, clear environment to an also beautiful but far more productive lake environment. And this is what we see. So here we've got stable isotope data for all 30 of those lakes. And you could almost imagine this as that Scottish loch biplot replicated 30 times here. And the first thing we learn is that it's very difficult to extract all of that same ecological level information from so much data. This is where I started to really learn data analyses which are specialized on stable isotope ecology. And that fills the second course, which you can take. Well, second of my courses, which you could take. And it highlights why those techniques, be they mixing models, food web metrics, why it's important to learn these or to understand these. Because we need to be able to make ecological sense out of all of this data. And when we do that, we can fast forward about 3 years of my life and look at this figure here. And along this figure here, on the x-axis, we've got what's a climate productivity gradient. Essentially, it's latitude. We're moving from the north here. As we move further south, the lakes get warmer and more productive. On the y-axis, we've got pelagic-derived contribution. How much, the degree to which pelagic zooplankton versus benthic, be it macrophytes or benthic algal production, is fueling the entire food web. Each of these points reflects the pelagic contribution to the entire fish community, scaled by mass, of a lake. And we've got red data and blue data. The red data reflects isotopes. So what we can see is if we look at our fishes, as the lakes get more productive, we're moving from almost 5% pelagic productivity, right the way up to some of these lakes we're getting 75, 80+% pelagic production, fueling the fish community. The blue lines represent stomach content. So here we've measured, with chemo and others, we also measured the stomach content of all of these fish and identified where they're feeding on pelagic or benthic prey sources. And what we see here, really interestingly, is that as we move along this gradient, they don't change. The fish community is consuming broadly equal amounts of benthic versus pelagic material, or at least where that varies, it's not driven by, or it's not related to productivity. Yeah. So we've got two very different signals coming from our data set here. The fish are feeding in the same habitats, but what's fueling their food has shifted completely as we've moved across this gradient. We've effectively moved from a characteristic clear state lake fueled by benthic algae. We've added some temperature and productivity, and we end up with this green state lake where pelagic phytoplankton driven production is dominant. Okay. That paper, I'll stick a link to that paper below. It's Ecology Letters several years ago at this point, 2014, I think, maybe even later. That's that story. That's the lake story. I also want to talk a little bit about some river work, and this is some river work that was done by myself and some others in beautiful New Brunswick during the years that I was there. And all of these pictures here are actually the area where I used to walk my dog every morning before heading into SynLab to play with isotopes. And as I was doing that walk every morning, I became increasingly aware of the strength of seasonal variability within these ecosystems. Let's start here in autumn or the fall, as it was there. Huge pulse of terrestrial material enters the river as all of these deciduous trees drop their leaves, and that leaf material is funneled down towards the river. Then everything freezes. We hit a period of 3-4 months where mean temperatures are well below zero, and that drives snow cover, ice cover, the lakes becoming closed, rivers are even becoming closed. Spring comes, the snow starts to melt, all of that material that's cascaded down onto the snow over winter is also becoming mobile and entering into the stream. We move on through spring, the ice is now all out. We start to see a lot of primary production in the stream because there's plenty of nutrients have just come in, but we've got full light access. Yeah, the canopy hasn't opened yet. Full summer, the canopy is opened, the river levels are starting to reduce. Again, different conditions, different reduced opportunity for primary production, certainly within the stream. And I was really interested. I've done work on this seasonal shifts in lakes. I was really curious as to how we could see this within the streams. And if you have a curious PI, then you need to have willing and equally curious lab members. And this is Bobby Nakamoto and Rachel Forbes, who certainly fit that bill. And Bobby and Rachel and some others and I spent a bit of time sampling rivers around New Brunswick through the seasons, through just after ice out, early in the spring and later in the fall. Well, spring, summer, fall, let's call it that. And really what we were trying to do was to develop a model, if we could, for how seasonal changes in the environment cascade up into stream food webs. And what we expected was that starting in winter, we'd have predominantly a terrestrial driven system with a lot of that CPU, coarse particulate matter, that fell in during the fall, breaking down to fine particulate matter, still allochthonous material fueling the food web. We then move into spring. We start to see greater pelagic or algal production. So this green component of our food web starts to increase. That maintains in through the summer. And then we move into fall or autumn, and we see that as the light decreases, that autotrophic production also diminishing, and another big pulse of allochthonous leaf litter enters the stream. What do we see? Here I'm just showing you the results of some mixing models which basically estimate the relative contribution of algal biofilm, leaf litter, and macrophytes to the benthic invertebrate community, all the invertebrates within those streams, replicated in fall, spring, and summer. And I should mention there's 2 separate streams here, probably around about 10 sites altogether, so quite well, reasonably replicated. And leaf litter is the dominant source throughout the seasons. This is a small stream, so Venote, tap your hat, because he got this one right as well. So we've got predominantly allochthonous fueled invertebrate community, but we've also got a lot of variability between seasons. This fall signal, by far the strongest evidence of association with leaf litter within the invertebrates. As we move into spring, macrophytes take on a bigger role as the canopy is open and we start to see macrophyte growth. And then as we move into the summer, things have stabilized out a bit, but certainly we've got the highest contribution of biofilm in summer. We can also, with our isotope data, we can also estimate food web area, food chain length, things like that. And that's when we apply those tools here. This is what we see. We see that particularly in the fall, when all that leaf litter comes in, it's increasing the spread of isotopic values within our invertebrate community. That's telling us that the spread of data, the spread of values, it's reflecting that greater diversity in production fueling the food web. And that's exactly what we're tracing down here, or what this is revealing to us. These are 2 cases. These are 2 case studies. I think I might make a few more of these little webinars highlighting a few other case studies, because this concept is applicable not just to these systems, it's applicable to all types of systems, be they freshwater, marine systems, terrestrial systems, anything you can get a sample of, we can do this with, okay? This means we can resolve food web and ecological function questions. If you're putting a dam in a river, if you're taking a dam out of a river, if you want to assess how something has affected the ecological function of a region, here's a tool with which we can do that. We can look at particular species, populations in great detail. Here we've got an example, and this is again published data from Finland, tracking ontogenetic dietary shift in perch, showing that larger perch are feeding on fishes relative to smaller fish, smaller perch, which are predominantly feeding on invertebrates. And crucially, or importantly, different sources of organic pollution coming into freshwater or marine systems carry with them tracers, isotopic tracers, which we can relate back to their initial sources. Yeah. And if we can do that, this gives us extra details and extra information with which to be able to answer this question, such as, my lake is polluted because of nitrate. Who's responsible? Where is that nitrate from? Isotopes can help you answer that question. I've been playing with these markers and working in this field for 20-something years. And I've done a lot with it. And I'm now transitioned almost fully away from my academic hats. I might still have some academic hair, but I certainly am wearing an academic hat very often. But what I am doing is finding new ways with which my understanding of these systems and knowledge and how to apply these systems can benefit other researchers, but also industry and commercial groups who are also challenged with or tasked with answering these ecological questions. So if there's a light bulb going off in your head right now, send me an email and let's get in touch and let's make something happen. If you're having difficulty with your own project or your own analyses, I'm more than happy to have a quick chat and get people back on the straight and narrow, let's say. I do offer a lot of training. I run group training. In fact, there's going to be some of that coming along pretty soon in a variety of locations. So watch this space. At the moment, I've got a couple of online courses which go through the theory of this and also the data analysis component of this. And this is the aforementioned Simon Johnson. Simon guarantees us that there will be an FBA stable isotope course coming along this autumn. So keep an eye out for that as well. And hopefully I will get to see you and chat some more about isotopes then. Yeah. Okay, that's pretty much what I told the good people in Edinburgh last week. So you're officially up to date. Not unlike then, I've got a minute or two over time, but I hope I've convinced you that isotopes are a useful tool, a powerful tool, something that you want to do. And if you want to do it, get in touch, because you can very easily and quickly bring my experience and my expertise into your project.